Go ahead, Madam Prorector.
Okay. Welcome, everybody. I hereby open this academic ceremony in which Ryan Alexander Miller will defend his academic thesis, Pathway Connections, Connectivity of Pathway Elements and Their Directions in Biological Molecular Pathway Diagrams. May I invite you to present a summary of your study and the conclusions of your thesis.
Thank you. Dear Prorector, members of the Corona, dear family, friends, and audience, in the next 15 minutes, I will shortly discuss my work entitled Pathway Connections, Connectivity of Pathway Elements, and Their Directions in Biological Molecular Pathway Diagrams. Biology itself is a complex process built from many different basic elements. And this work actually focuses on biological processes and how they form complex networks from these basic elements. These elements end up being our building blocks towards a biological system. And we can then draw this to create a representation for the elements of our system. So in many ways, this will be analogous to a street map. Say we want to go from our home to the park and then back home again. One would assume that we can just do this, but not so fast. Much the same way that biology has rules, we also have traffic rules as well, right? Some streets you can only travel one direction, other streets you can travel both directions. So instead, what we might do is we might leave our house, go around the corner, and then follow the street down to the park. And then play some football in the park, go back around, and then when we're ready to return home, we go follow around the park, and then back up the street, and we can return home again. These directions, these street directions, are an analogy to how biological processes also have to follow these rules of how things are connected to one another. So the basic interactions between elements provide the basis for our system. And in biology, we call this system an organism. So whether that's a redwood tree, that's a wolf, fungi, or microorganisms, they're all this complex system built from these basic building blocks for a system. These basic elements, there's two of them. One is going to be the nodes, the nodes that connect things. These will be things like proteins, metabolites, and genetic elements. Genetic elements being things like microRNAs or small interfering RNAs. So, for example, we have things like, these are our nodes, right? These are our proteins, PI3K, AKT, TSC complex. But then to describe the picture and to describe how the system works together, we also have to know the edges or the connections between the nodes or our basic elements, right? Our building blocks, right? In this case, we have an influence from the PI3K, which is going to influence another protein, right? And it moves in that one direction. And then we have AKT that's actually inhibiting the action of the TSC complex, right? So this is actually how we collect the knowledge of how the things influence each other. And with this collective knowledge, we can do things like study how two drugs might actually work better together than individually. It allows us to do things like identify alternative protein targets, and allows us to do things like study rare diseases. So first, we need to actually model how these diagrams actually represent biology, right? So the lines actually need to have some sort of meaning that connects one element to another in our pathway diagrams. And in this case, we have something like gene A will influence gene B. This is some sort of directed interaction. And we have gene B will influence gene C. And we can have things like complex interactions where maybe there's some sort of binding event where they actually share the connections. There's no implicit direction assigned to that interaction, but it's still part of the system. So for a resource like Wiki Pathways, we would store this information as RDF, database information, and we actually explicitly store the connectivity information as point A is connected to point B. Now, these connections can be either directed or undirected, but to a human that's actually looking at this pathway, it's fairly obvious what an arrow that connects 2 points means. But for the RDF or for the machine readability, we need to explicitly state what these connections are to one another. And then we also, on top of that, there are different ways to draw the same interactions. So we need some sort of harmonization efforts in order to make sure that notation for one interaction type is the same as the notation for another interaction type. So we can use that modeled data. We can implement it, this pathway and this connectivity information into platforms like OpenFacts. OpenFacts is actually a drug discovery platform that uses linked data approaches to connect resources like KEBI and Kemble, Uniprot, and Wiki Pathways. And this work actually added the access to specific calls to up and downstream interaction effects. And this actually allows us to do things like identify alternative targets using the OpenFacts biology and chemistry data with our pathway data that we want to use. So, for example, below we have part of the bone morphographic protein signaling pathway. And if we actually want to target BMP receptor 1, we see that maybe we can't, it's a hard target to target directly. So we look to see, are there things nearby that we would say might have similar effects to it? We look and we see, the first thing that we see is something like BMP2 that's nearby. And then we say, well, how do we know that BMP2 might actually be a good thing, a good target to look at, alternative target to look at? We actually look at these connections, right? And we see that indeed BMP2 is connected to BMP receptor 1, and this means that the BMP2 has some sort of influence on the receptor. This work then can go into other areas as well. Other areas where this is useful, something like subnetworks in rare diseases. So we can do things like we can create a novel subnetwork for a disease state using Wiki Pathways connectivity. For this example, we were actually using Rett syndrome. And I used data retrieved from post-mortem Rett patients from brain tissue samples. Then we do something like creating a large network using human pathways from Wiki Pathways. And we actually use these commonalities that connect one pathway to the next pathway in order to create one huge pathway or one huge network that represents all of the pathways that represents a human. We do have to remove some extraneous metabolites, things like ATP, ADP, NADH. These things end up connecting everything to everything else as their energy pathways or energy mechanisms. And what we end up with is a very large network, over 900 pathways, tens of thousands of nodes and hundreds of thousands of edges. It just looks like a giant hairball. But what we want to identify an active subnetwork within that for that disease. So, for example, this is actually one of these active subnetworks from the brain tissues, where we can actually see that the rounded rectangles actually indicates significantly changed genes, and with the blue color indicating downregulation and the red color indicating upregulation of genes. This work can also be used in areas like cancer research. So drugs themselves are often developed as monotherapies for treatment. But sometimes the combinations of two or more drugs are actually more effective than an individual drug, and the effects, when the effects have an effect greater than the sum of the two combinations, we call this synergy. The combinations themselves can be synergistic, they can be additive, or they can be antagonistic towards each other. And then using connectivity data, we can actually suggest combinations that have the possibility to be synergistic, rather than simply taking all combinatorial combinations of drug combinations. So we have a couple different approaches that we can implement here. One being this idea that two drugs might share the same target. This is the resulting network that we get with that. Another approach would be that using this information, can two drugs share the same pathway or same or a similar biological process or ontology? Or another approach could be to completely disregard the pathway boundaries themselves or how people drew them. Instead, we use individual connections and say that simply that two drugs for this combination need to share a common route through a pathway or a common route through our network in order to potentially be synergistic with one another. So the understanding of connectivity and directionality actually helps us to understand biology itself better. So whether that's implementing things like drug repositioning or drug repurposing, things like identifying active subnetworks within a larger disease state or rare diseases. Or we can also use it to identify potential drug synergies between effects. These all actually help us to leverage our understanding and prior knowledge in biology and how things are connected to each other, how the system should work together, and how we can use this information to help us actually understand biology. So I would like to thank you for your attention, and I will actually now give the floor back to the prorector.
Thank you very much for your presentation. We will now start with the opposition, and the opposition will be opened by Professor van Schoten. Professor van Schoten was chair of the assessment committee, and he is professor of genetic toxicology at this university. Professor van Schoten.
Thank you. Dear candidate, compliments for the nice dissertation, from which there is a lot to learn and therefore very interesting. Also compliments for your promotion team. I'm a biologist and biology is complex, so I'm used to complex materials. And I have to say that reading your thesis sometimes made me dizzy from the complicated nature. So it was a challenge for me to read, but it gives me all the more material to ask questions. Just to start off the discussion, can you read for me proposition 1? Do you have the propositions with you?
I don't have them immediately in front.
Okay, okay, I can read it.
Yeah.
Okay. Networks are found all around us. So I was puzzled by this proposition, and actually I wondered, what do you mean with this, and what's the implication of it?
Yes. Thank you for your question, highly esteemed opponent. Networks are all around us. They could be simple things like computer networks. They can be biological networks. They can be ecological networks, right? We're literally surrounded by networks. We're also internally we're composed of these networks. That was one of the portions, the points of my work. So yes, absolutely, they are all around us, and in us, and and everything.
Okay, so can you also then use your models to describe other networks, like for instance social networks?
Fundamentally, the principles should be the same. One connection to another, or as long as you have some sort of way to describe these interactions between elements of your network, so in between people, whether that's stopping someone from posting something or influencing other people to post things or read it, this should be, these principles should apply to.
Okay, interesting. For me as a genetic toxicologist, chapter 6 was very interesting. And in this chapter, you are searching for synergistic drug combinations for cancer treatments. And that's very relevant because these combinations could potentially overcome treatment resistance when using a single drug. So on page 123, you have a table 6.1. And can you explain that table for me?
Yes, absolutely. In this table, we actually have these are percentage of cell viability for two individual agents, agent 1 and agent 2 being something that some sort of drug that's targeting HKT protein, and the other one that's targeting ADAM17. So the top row and the first column to the left are the concentrations of the concentrations of these drugs, and the second row and the second column are actually the cell viability after treatment for those individual.
Okay, so what does NA means? That means not applicable, or?
Yeah. This is exactly how the data was presented to us.
Okay.
Yeah.
So that's what you want to try to predict.
Yes, absolutely, because these are actually monotherapies that we predict. So many of these cancer cell line cells will die under these conditions. But yes, we're trying to predict what the combination, whether the combination of AKT and ADAM17.
Okay. So I see here also a dose range of the drugs. So I wondered, is it also possible for you to implement the dose-response relationship, because that's quite important for drug synergy assessment. Is that possible? You did not do that, right?
No, I used a simple model for well-described model called the Lewis additivity model. As this mathematical model in the background, I was instead picking combinations that, for whatever reason, seem to make sense as combinations to work with one another, and then using that model to make our final call as to whether something is synergistic or not. But yes, there are, of course, there are methods to incorporate dose response.
Oh, that's possible.
Yeah,
it will be possible in principle. So maybe that's a next step you can take.
Absolutely. That's an interesting way to take it, and actually it's an important part of the story, right?
Okay. To go on further, cancer is a disease with multiple mutated genes and several pathways that enhance proliferation are involved. And as I understood from this chapter, you are looking in one pathway. And my question would be, is it possible to go beyond pathways, that you are, for instance, looking at pathways that are not connected with each other?
Yes, one of the approaches actually was to disregard pathway boundaries or the way they're drawn, and instead use connections from one element to the next element, right? And simply having so many steps in between each element, and saying, but they still follow a common path. So whether they're part of two different pathways, these connections, we still have information about, PI3K is connected to AKT, right? We still,
Okay.
I mean, these a lot of times these processes are kinases particularly are going to be present in all sorts of pathways, right? So
Yeah.
Yeah, we can absolutely disregard just the pathway information, instead say they just have to have a common root. If we target the root at two different places, hopefully we can circumvent resistance.
Okay, because I read somewhere that the targets cannot be more than 4 intermediate steps apart from each other. Is that right?
In this case, yes, it was 4 intermediate steps. We can use other numbers. We can use 3. We can use 5. 4 seems to be a good number. Once you allow too many steps in between, so it can be 4 or less, or 5 or less, or 3 or less, right? Once you allow so many steps in between, you can actually eventually start connecting everything to everything, and then you're taking literally every combination possible, right?
Okay, yeah, I can see that. Then that makes no sense. Do I have more time, Prorector? 1 minute? Okay, you're muted, but okay, just.
Yeah, now it's only half a minute, so.
Oh, half a minute. No, I just wondered, you found indeed many combinations at a certain point, and then my question would be how you can validate this and how what's the next step. But maybe that's a discussion too long, and we will do that when we have more time. Thank you. I'm satisfied with the answers, and I give the word back to the Prorector. Thank you so much.
Thank you very much, Professor van Schoten. The opposition will be continued by Dr. Feite. Dr. Feite is assistant professor at Maastricht Clinic at this university. Dr. Feite.
Dear candidate, congratulations with this thesis. I actually really enjoyed reading it. Also, congratulations to your promotion team, obviously. I really liked that you had a lot of technical papers in there, but also a lot of applicable things, right? You actually answer a biological research question. with your findings, which I always really like. So well done on that part. I have a few questions, mostly related to chapter 3, and also a little bit the connection with chapter 2. So it's going to be a bit more technical than the previous opponent, but I'm sure you'll do fine. First question I had was, it kind of refers to figure 2.1 from chapter 2, which is where you show the RDF network, right, for the pathways. And then there is one RDF type, or let's say a line type, that I found interesting, which was called the directed interaction, I think. And I have a question about that one, also in relation to chapter 3, because when I was reading chapter 2, I was like, okay, interesting, because with RDF, usually there is no directionality, correct, right? It's a knowledge graph, but there is no direction given.
Yeah.
But you have this interaction type there, right? It's a directed interaction type. So I was at first curious about how you did that. And then I went to chapter 3 and saw that you actually use checks on top of the RDF to infer directionality, right? So that confused me slightly. I think I know why you did it and stuff, but I would like to get your, to pick your brain a little bit about, okay, how do these things relate? Do you use both of them? What's the purpose of the directed interaction, for example? Can you shed some light on that?
Absolutely. Thank you, esteemed opponent, for your question. Again, the way RDF allows you to, the fundamentals of it actually allow you to give semantic or metadata or background information about anything, right? So this includes things like interactions, right? So we can actually say that when someone draws an arrow, a line with an arrowhead on it that connects, one node to another node, we could say that this is some sort of directed interaction. We can also store specific interaction information within there as well, whether that direction is a result of something like a stimulation event or an inhibition, right? These are all different types of directed interactions that we can use to put in there. And the checks, yes, the checks was included for it stands for these shape expressions, right? So we're actually describing what the shape of particular interactions should look like, whether those are two elements that work together in order to catalyze some sort of reaction or any number of things like that, right? But these can also be directed as well. So yeah, it actually describing the shape of what's allowed is actually useful for the modeling of the RDF.
So if you have this directed interaction, right? How do you know which way the arrow points? Does checks do that, or is that inside?
No. That is actually handled by a JavaScript that actually takes our data and says that one end, if we know that it's a directed interaction, we know that one end will have an arrowhead on it.
Mm-hmm.
Right? We'll have an arrow pointed towards it. And when we have this, the end of the arrowhead, we know that's where the thing that's being influenced is so that we know the direction of that the arrow is pointing that way, right?
But you can also extract pathways in RDF format, right? So what would that look like with the let's say we have two genes, right? One influences the other. So is then the one that influences the other the subject and the other one is the object, and that's how you know it, or is that not possible to figure out in RDF and you need to go to PathPhysio for that?
Yeah, the RDF actually has explicit interaction subjects with.
All right, so the gene is like the subject, and then it has it. Okay,
And then it
but
has an
then
object.
yeah,
Then it
yeah,
has the object of these two endpoints, right?
right. So then the object is the one that the arrow points to, I guess.
Yes.
Yeah. Okay, that makes sense. Yeah. That was a little bit confusing for me when I was reading your thesis, but that really helps. So about these RDF pathways, right? I know there is this RDF transformation tool that transforms RDF, right? I think is also mentioned in chapter 2. I was just curious, are these, which pathways are available in RDF format? Are those only the curated set of pathways, or are also uncurated pathways available in RDF?
At the time of the paper, it was only the curated collection that's automatically created into RDF. You could actually use the script against a local instance, just telling it which things to transform into the RDF. But in this case, the thing that's done automatically is the wiki pathways, curated collection, the Reactome collections, and then some NetPath collection and stuff like that as well. So
right. So that means that if I were to make a pathway and I want to make it into an RDF format, that's something I have to do manually, I guess, or with a converter or something.
Yes, exactly. You'd have to use the script that is available on GitHub, but yeah, you'd have to implement this
Right.
the script.
Interesting. Follow-up question on chapter 3. I looked at tables, let me see, 3.2 and 3.3, which is where you show an overview, I believe, of the numbers of nodes and edges, or the data node type, and then the counts for 2 types of formats, correct?
Yeah.
And first, can you maybe explain what the difference is between the 2 tables? Oh, it's interaction type and data node type. Yeah.
Yeah. So we have table 3.3 and which one?
3.2, right? So
Okay.
the 2 that showed the counts, basically.
Yeah.
3.2 is on page 54, and the other one is on page 57.
Yeah. Right here. Okay. So the the 2 different types are actually the wiki pathways RDF. This is the thing that that stores this semantic information about how elements are connected to one another, right? This is connected to that, that's connected to this, right? A, B, C. The GPML actually is the graphical representation. So we store that information separately, but for completeness, it is also there as well. And the difference between the tables is actually the counts for the data node types versus the interaction types, right? So one is how many metabolites, how many proteins, how many pathways, are we counting? And then another one is how many of the interactions do we have, right? How many directed, how many indirected, how many conversions, how many catalysts? Yeah.
So I think I need to round up. I have one small question before we stop. I was very intrigued by the fact that these counts are so different. So I was trying to figure out also what's the difference between the 2 types, and you've slightly already told me that, but can you briefly maybe explain why the counts are so different, right? Because even if you look at, for example, proteins, like the one has 8,000 proteins and the other one has 140, right? That's a big difference. So I'm just curious why.
Yeah, the semantic layer is the layer that actually has the more formal rules around it. So we actually curate all this information, things like this. But we can actually have things like, if you actually look at the interaction table, there's this count of unknown interactions that are only found in the GPML RDF. This could come from things like how people actually draw the diagrams, right? Did they actually connect the lines, or is there like a little bit of space and the script didn't quite know that they were connected to one another? So things can drop out, from that based on that. So we can actually lose some information based on user information. We try to connect this as much as possible through curation efforts, but yeah, it can be quite difficult sometimes.
Okay, thank you. I give the word back to the prorector.
Thank you, Dr. Feiter. The opposition will now be continued by Professor Schneider. Professor Schneider is Professor of Bioinformatics at the Université du Luxembourg in Luxembourg.
Okay, thank you very much. Yeah, dear candidate, thank you for the work. It was a pleasure to read. Let me start with my first question. Did you look at post-translational modifications? Because sometimes the react. Did you try to incorporate it?
Professor, I think I got a hiccup and I missed part of the end of your question.
The question was if you tried to include post-translational modifications, like phosphorylations and these kind of things.
Dear highly esteemed opponent, thank you for your question. No, I haven't taken into account post-translation effects. This could be, again, this is all metadata though, so I mean, we can add information about, this, that a protein might have a different state, right? Because it's, or a gene might have a different state because, like you said, it's been methylated or something like this, right? So.
Okay, then I just jump in between the chapters. The MECP2 protein and the Rett syndrome. Do you know how this is inherited? Or if?
MECP2 is like, it's an inherited trait found on the X chromosome. Yes, so this is why these patients actually are all female, right? Males actually don't survive very long with this condition. So it ends up being a developmental disease that actually affects normal biological development. And
actually, it's yeah. Interestingly, it's not inherited. More than 95% are sporadic with mutations in that gene. So that's okay. That's the detail. Then in chapter 6, when you do these, you call themselves hairballs. That's especially when you increase the networks or when you look at drugs and proteins in one network, in one graph. Did you look at methods which try to get a better visualization done?
Yes, but visualizing them wasn't necessarily the end goal. Instead, we were just trying to capture the combinations. The visualization was done as an aid, actually, but yes, these hairballs can be quite difficult to read, difficult to understand actually what's going on within them. So there are, there are tools that you can use to sort of rearrange things so that they the connections are stayed, but they're spaced out a little differently, and connections are preserved in that way. like I said, the focus wasn't necessarily on making those visualizations more readable. Instead, the focus was on the actual combinations that we were after.
But how do you then read out the interesting parts? Which drugs, which proteins, if you have this hairball and that is unreadable? So do you have methods or scripts at hand where you can filter it?
Yes, absolutely. As part of one of the GitHub repositories, there actually are RDF calls that actually allows us to access our pathway database. You know, all that background information that we have about how things are connected to one another. Yeah, that's where you can actually get this information, right? There is a script. This script actually uses the RDF, like I just was discussing earlier, that gene A is connected to gene B via this arrowhead, right? And we explicitly state every single interaction that we have, right? So this is how we know that A is connected to B, B is connected to C, C is connected to D, all the way down through. And this is how we can actually make our connection. But we also store information about which pathways they're also part of as well. So we do actually have all of this semantic information in the background. It's just finding a way to leverage it, leverage our knowledge. We actually have there's so much knowledge that's actually being lost right now, right? Because people are doing all this work, they're doing all these descriptions, but there's a real need to actually find a way to leverage this information that we have.
Mm-hmm. In one graph, you also had over and under expression, I think, of genes. Have you thought about how you could model this in future? You know, dynamically trying to find out what happens in this network now? Do you know methods about it?
Yes, I mean there would be, you could conceivably add again with something like Cytoscape, you can add information to these edges, right? About fluxes and things like this, right? So we can actually give some sort of weight to the interaction, saying that a reaction is really inhibits this other protein, right?
Mm-hmm. Okay, and then for your trucks, in your examples, we have the two trucks hitting same pathways or same targets in principle. Do you know how many trucks are on the market which have a multi-target actually? They hit different pathways, not even related ones?
I do not know off the top of my head, but I do know that RF target and side effects are definitely something that you have to weigh against this. And if you're targeting multiple pathways, you might also have multiple side effects as well, right?
Mm-hmm. Yeah, exactly. Okay, thanks.
Pro-Rector, I think you're muted.
The opposition will now be continued by Dr. Maran. He's principal IT business partner at AstraZeneca in the UK. Dr. Maran.
Thank you, Proter. Candidate, I would just like to congratulate you on your thesis. Like many of my colleagues on this call, it was a pleasant read, if a complex one, and did challenge my mental torture at one point of trying to join some of the dots together, as you've outlined in chapter 4. Yeah. I would like to open the questioning, focusing on chapter 4 and following on from a number of the questions that have already been asked. You chose wiki pathways and you chose OpenFacts as collected data sources. You've already outlined that you can end up with a hairball as a result of the interaction map that is ensued. how did you go about limiting that dynamic hairball in a way that it became manageable?
Dear esteemed opponent, thank you for your question. Yeah, that's one of the problems with these networks is they very quickly can get out of hand, and so we do have to sometimes put limits on how far away we can go. Again, there was, I discussed earlier that once you start adding elements in between pathways, you can end up connecting everything to everything else again, and then we just end up with this hairball again, right? These very complex networks. So we do have to put limitations on how far away we can go, whether that's one step, that's two steps. In the case of open facts, we can actually chain calls together to say, like for example, the BMP receptor complex. We can chain that with a call and say what's upstream of that, and we get BMP2. And then we can say, okay, what's upstream of that? We could say that NOG is inhibiting BMP2, right? So we can chain calls together to create as complex as we want. But the basic element is that, it's only one interaction at a time in that case.
So just following on from your answer then, so your assumption is that there is one interaction, but we know that in biology, nothing is straightforward. And we as intellectuals tend to disagree about what the interaction is real and which is not real. So how do you accommodate the intellectual disagreement when you have an RDF saying it's directional? Is there a way you've addressed it or thought of addressing it?
The only way that I can think of off the top of my head would be we do curation efforts to, where biologists actually sit down and make curate a pathway, right? Fundamentally, Wiki Pathways is a wiki, right? So it allows users to create their own pathways, but we do have expert curation on those pathways to control for the quality, to control, we can more reasonably agree that maybe they are connected to each other and they have some sort of influence on each other, right? But we also allow for things like simply saying that an interaction is directed towards another element without regard to what the specific interaction is, because that does come up sometimes. We know there's some sort of interaction between these two elements. We don't know exactly what it is, right? We do allow people to draw that and represent that in Wiki Pathways. This is also captured by the modeling of the RDF data.
So with that in mind, have you considered using a weighting algorithm based on intellectual assertion? So if three intellectuals say this is an appropriate interaction and one says no, you give it a sizing or an arrow width, if you like, in terms of the effectiveness of the interaction?
Yes, I mean, again, this is fundamentally possible. The RDF allows us to capture any sort of metadata about the interaction that we want, right? One way we can go about doing this is we can add, say we have citations on an interaction. The more citations that we have about that interaction, the stronger the evidence is that this is indeed the interaction that we're looking for and describing. So it should be possible to add some sort of weighting to these interaction effects.
So with that weighting, do you think that is a way of providing understanding to the rare disease angle that you proposed in your thesis?
Yeah, absolutely. the rare disease angle, although the reason we choose rare diseases is because oftentimes things that are common, say something like colorectal cancer or breast cancer or something like this, there's actually oftentimes there's someone who will specifically map out that disease state, right? Instead, we can quickly identify which elements from a human network, right, from all of the human pathways would be implicated in this rare disease, right? It's sort of a quick and dirty way in order to identify things that we don't explicitly have to draw out all of this pathway, all of the elements that are interesting to us, right? But absolutely, adding some sort of weighted evidence to it would definitely make it give people confidence, right? That when you have one element connected to another element, we have six citations that describe this this interaction effect, right? This inhibits this. We have this evidence. We have a bunch of evidence. Here's another line. We have no evidence, right? This one would be more interesting, right? Maybe more interesting than the other, right?
So let me probe just a little bit further. So have you considered doing the inverse of that, looking for the rare outlier? You mentioned multi-drug therapy and potentially reuse of drugs.
Yeah.
One thought would be, reading your thesis, that there's an opportunity to look at it from the inverse model, where you're looking for the outlier of a disease which cites a particular pathway that's never been discovered before, and looking for a new monotherapy or a dual therapy. Is that something that came up in your research?
That actually is outside of the scope of what I did, but it is an interesting perspective. I hadn't previously thought of that angle, right? Instead, it was we were participating in originally it started because we were participating in a project that was actually trying to predict drug synergies, right? So,
Sure.
but in theory, it should be the same as well, right? If we could say what processes are targeted, we could say what processes are not being targeted, right? And get a list of processes or pathways or whatever that aren't being covered or affected by this gene. That also chemistry data that we have, from places like OpenFacts, things like this, right? We do have information about what their targets are, right? We also have information about, off-target effects, right? Side effects that might come from it, right? Yeah, I think leveraging that information is interesting. It should be possible to do the inverse, even though that wasn't necessarily what I did in my case.
Understood that. One last question, if I may. So,
Very short, please.
Yes, I will do. Well, so your data sources that you used, when you were looking at them, was there a surprise to you about the limitations?
I wouldn't say surprised, but yeah, of course there's limitations. Sometimes also the limitations oftentimes come from actually comes down to how people actually draw a pathway diagram, right? Whether they actually connect all the elements to one another. So that can be surprising in the sense that, there's the graphical information says that there's a line here, but it's not connected to anything, and so that's not represented as an interaction in the semantic layer, right? So we actually do lose information that way. We try to minimize that, obviously, through curation efforts, but you can't catch everything, right? You do the best you can to, and we develop these scripts as well to help, identify, this isn't completely connected, this is a line, should it be called something else, right? Yeah.
Thank you. Proctor, thank you very much. I'm finished with my questions.
Thank you very much. Then we already have the last opponent, Professor van den Hoven. He's Professor of Neuroepigenetics at Maastricht University. Professor van den Hoven.
Thank you very much. Dear candidate, first of all, my compliments on your very nice thesis. Also my compliments to your promotion team. I actually first had a question on your presentation. You were referring to playing football in the park. I was wondering, considering the discussion that Joe Biden had this week with Mark Rutte, whether you were referring to Joe Biden's definition of football or Mark Rutte's. I'm assuming the first one, but okay, just kidding. Let's leave that out for now. My first question is actually on all my questions on chapter 5. And the first one is on the degree of expression related to the changes in RNA expression. So at the one hand, you take that into account. I think you use a cutoff of 1.5 for the fold change,
Yes.
but you do not actually use the absolute difference in expression there. Could you comment on why including it or incorporating in one way or another, but not completely?
Dear highly esteemed opponent, thank you for your question. Actually, I did tailor that towards a European audience, so I actually meant European football to help you out there. Plus, we have the World Cup going on, right? So that's also fun. But yeah, when the decision was made to use these fold changes to determine what's the cutoff for what we consider a significantly changed gene, right? So we have to pick some number, right? It comes from expertise that we picked this 1.5 log fold change as being a significant increase, and so you can't use absolute values because different genes or proteins are going to be expressed differently, right? So you need some sort of way to account for different amounts of things that are there, right?
Yeah, I agree on the latter. For the first one, of course, you can base yourself on statistics. So a 10% difference in expression could still be biologically relevant and significant. So the statistical significance could also be used instead of the cutoff value of 1.5, although a lot of people use that. So it's not necessarily criticism. Okay, thanks for that. And maybe I will skip my second question and move to MECP2 that was already touched upon by one of my colleagues. Yeah. Yeah, it's indeed not surprising, the findings that you do, particularly at the level of transcription, where you say, hey, this really adds something to the wiki pathway information that is out there. At the same time, this could really be expected, of course. This is a regulator of epigenetics, of DNA methylation. Yeah. And you actually also refer to some other papers that already addressed and found this before, one of those publications actually being of your group members. I was wondering then, if previous analysis already hinted at those mechanisms being involved, transcription, etc., what is then really the added value of your approach that integrates these gene expression data with the wiki pathways? Maybe you could briefly comment on it.
Yeah, absolutely. So those we picked Rett syndrome and MECP2 because we actually did have some expertise with some colleagues. That was about developing this approach where we could identify active small networks from a larger network. We actually used that information to validate whether our networks even made sense, right? So there was some surprising, effects, some proteins that affect, liver function and things like this as well. But these were also have been discussed in the citations and that my colleagues have wrote. So yeah, this is possible. Yeah.
Cora asked.
I give the word back then to the prorector. Thank you very much for your answers.
Thank you very much. Ryan Miller, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense, and I request that you and your company await the results of our deliberation and our return.
Thank you.
We are here, please unmute.
We're back, and Ryan Miller, the degree committee here present online, has assessed the quality of your thesis and of your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Evenlo is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Thank you, Prorector. Dear Ryan, before I start, I have to ask you a very serious question and ask you to make a promise towards the society and this university. And I will read out that promise to you first. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Thank you. Then, by the authority vested in us by law, and in conformity with the decision of the committee here present online, I hereby confer upon you, Ryan Alexander Miller, the degree of doctor, and grant you all the rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university, but it will actually be shown by the beadle.
Thank you.
Thank you.
The word is now to Dr. Willighagen for the laudatio.
Dear Ryan, dear Dr. Miller, it is a great pleasure to be able to congratulate you here with your degree. I'm going to speak from a letter. I'm really happy that we are here. We had a long time together. You came from the United States. You joined our group in 2015, working on the Open Facts project. We got to know each other, both working on this. I, as a postdoc at the time, well, at least at the start, when I joined the group as a postdoc, I started working on Open Facts. And I wasn't actually your supervisor from the start, and I only got to know you along your way. First in the Dream Challenge, which was a project that Chris picked up, an international challenge to the drug synergy here. In this case, they have many challenges, and Chris came up with that idea. We wrote a proposal, and you and Linda, a colleague at the time, you worked with students to solve this synergy problem. And you came up with a lot of interesting ideas. So a bit later, when during your project, when Chris asked me if I could become your daily supervisor, I was quite happy to do it. I've known you as a friendly, open guy, and it has been a great pleasure to work with you. After that moment where I picked it up, we worked on a number of papers. You really worked hard for that. Some of the things were really complicated. During the defense, we saw some of those things, the lack of information, the need for harmonization. And you picked up these things one by one. It wasn't always easy, but along the way, we got further and further. And I'm really happy how things went this way. At some point, your contract ended, and that is actually, I think, a moment where our collaboration changed quite a bit, because the supervision meetings actually became online, and at that moment I got to know you even better, because at those moments we talked about the papers itself, but also we talked about other things. Soccer, not so much really, but about other things, the things that you like to do, the things that you were interested in, and unfortunately also a very interesting, challenging presidency at the time. I really enjoyed these meetings that we had together where we were finalizing these things, and I'm particularly happy with this chapter about modeling the interactions, which was perhaps not as clear from the defense here, but is really an essential part in the Wiki Pathways project, where this directionality, where the meaning of these things are being used now by in other projects to take advantage of that. Something that you did with the Synergy paper and with discovery of these modules in these networks. I think you did an amazing job. You showed great determination, and I think I'll end my congratulations with that. I'm really proud of you, seeing you standing here defending your story, and even more about the determination that you showed during this project. In the preparation of these of this meeting, we talked about how to prepare for a defense, and I think you actually did a lot better than me giving this laudatio right now. So I'm going to end now. Congratulations, Ryan, Dr. Ryan Miller, and it has been a great pleasure to supervise you. Thank you.
Thank you very much, Egon.
Dear Dr. Miller, also on behalf of Maastricht University, I'd like to congratulate you with the degree you have acquired. It's a pity you couldn't be here in person, but this is also a good way to finalize a long period of research. And I hope you have enjoyed the session and enjoy the rest of the day, because for you it will still be early, I guess. And of course, I also want to congratulate your family, your friends, your colleagues with whom you worked here and probably elsewhere. I'd like to congratulate your supervising team, Professor Evelo and Dr. Willighagen, with this PhD degree that you obtained today. I also like to thank all the members of the Corona that were present today, especially those from outside Maastricht, Professor Schneider and Dr. Marren. Thank you very much. The university really appreciates your contributions to our PhD defenses. And with that, I wish you all a pleasant day, and I hereby declare this ceremony